Okay. Well, you guys are having a little arts and crafts time. We're gonna talk about muscle fiber types, okay? So before I jump into this, I wanna revisit one thing from last Friday. If only because I promised you guys it would show up on the test. We're gonna talk about it again, okay? Again. Who remembers? Okay? I should have put the thing up so you guys can text in and we can take a poll. I'll have to remember to do that next time. Where in the cross-bridge cycle do we actually use energy? Where do we use ATP? I'm not saying this is the single most important thing that we will learn in class, but it's close. But it's close. Okay, wait, I got a hand, I got one over here so I can see. Kindle.
Use it to turn off, muscle.
Turn off the muscle. You're like 98% correct. Okay, you can be a little bit more specific about what exactly is happening to turn off the muscle.
When, it will reach out.
Okay, we're close, Okay, I got another cake. So we're hydrolyzing ATP, that's right, that's how we're actually using it, but what happens before that medicine?
When the myosin attachment.
Boom, got it. Okay? Myosin attaches from actin, we hydrolyze the ATP, and that, as Kendall said, right, shuts off the muscle contraction. Okay? We use ATP to relax a muscle. Use ATP to relax a muscle. Completely by itself. That's what we do. Okay. Good. Good. Let's talk about fiber types. Have you guys heard of fiber types before? Some of y'all are nodding, yes, no, maybe. Okay, somebody throw out a fiber type for me. 2x, okay? What the hell is, what is 2? Is there a 1? Is there a Z? Like what does the 2 mean? I don't know who said that, I'm sorry. Was that Lexi? Did you? Who was?
It's, like a fast switch.
Okay, you are 100% correct. 2x is a real thing. Fast switch is a thing. But what does it mean? What does fast mean? Why is it fast? What makes it fast? Okay, but what is fast? You're correct. Glycolytics describing metabolism, but what makes it fast? Cooper? It can hydrolyze ATP faster. Why can't it hydrolyze ATP faster? You're correct. Go ahead. It's the enzyme, it's the myosin ATPase. Okay, why did I make us talk about what we use ATP for first? What? The fast is from this myosin ATPase. What does fast enzyme do? If it breaks down the ATP, what's that leading us to? Use ATP to have a muscle do what? Relax. Fast muscles or fast twitch fibers relax faster than slow twitch. Okay, they also can turn the force on faster, but they relax faster. Okay, so again, all these things are tied together. We call it fast, we call it slow, we're talking about the metabolic profile of these things, but it comes back to there's something anatomically that is different about these fibers, and it changes their actual observable function. And that is why, right? You got a fast twitch muscle, or you have more fast twitch fibers, you run faster. To run fast, you have to turn the motors on fast, but you also have to turn it off fast, so you can do it again and again and again. Okay? So we're gonna get into that. That's where some of this comes from. Okay? We know about muscle fiber types because we were able to take a piece or a chunk out of your muscle and look at it under the microscope. Okay? Now, technically, we can do this here. If you want to come over to the department, we can dig around in a cabinet and find some biopsy needles and do something. We don't do this very often anymore. We did this, unfortunately, when I was a PhD student in my lab, a lot. And some poor grad student has to look through the microscope and circle the fibers and figure out what fucking color they are and characterize them and like, okay, this was a 2A, this was a 2X, this was some weird thing where it didn't work. That was me. Totally not bitter about that at all. Okay, but what you see here is the idea of taking a muscle biopsy. You make a small incision in your muscle. We insert this special needle in called a Bergstrom biopsy needle, and it goes in and it has that little window on it. And that window functions like a guillotine. But you open the window, you apply suction on one end and it pulls some fibers into that window. And then you drop the plunger or the guillotine and it cuts off those fibers. And when it pegs out, is this little piece of muscle. It looks like the eraser on a pencil. Okay. And again, some poor grad student pulls that thing out. We would mount it on a popsicle stick. Okay. Dip it in liquid nitrogen and flash freeze it. And then you put it in the world's most expensive W size you've ever seen and cut these tiny little sections off of it and look at them under the microscope. Okay. So we knew that muscles came in, their fibers came in different flavors or whatever from animals. But it wasn't until we could do this that we began. This was in the 1960s. Which I know seems like the dark ages to you guys. But it's not that long ago in the lifespan of science until we could do actual biopsies like this. Okay? Because I mean, none of you want me to let me cut out a big piece of your muscle and then go through there and tease it out and do something, right? That would seem bad. So we needed a way to take small chunks. And once we did that, we could then begin to see, do human muscles or human muscle fibers look like what we saw and observed in animals? Why will this not go along? Okay. So once we had these things, once we have the individual fibers, to look at the differences, we have to put different kinds of stain on those fibers. And based upon the differences, the function of different enzymes, the metabolic profiles in those, they will stain different colors. And once you see them as being different colors, you can go through and you can count up which ones are going to look this way, which ones look the other way. And then we can figure out in you, how many type 2 fibers do you have? How many type 1 fibers do you have? Okay. So the old or original way of doing this was something called histochemistry. Okay. Histochemistry. And this involved looking at the various enzyme functions. Okay. The function of the myosin ATPase, or the thing that is going to hydrolyze the ATP and let us relax our muscle really quickly. Okay. But you would take the fiber, you would incubate it with ATP or different other kind of substrates. And we would look at the myosin ATPase function. We look at the metabolic enzymes in the Krebs cycle specifically, okay, or in glycolysis, things that are in the mitochondria or in the cytoplasm to figure out to what extent can this fiber make ATP in the mitochondria, to what extent might it make it in glycolysis. And then we could also stain for how much energy substrate, how much glycogen does it have? How much intramyocellular fat does it have? And from that, you can begin to get a little bit of a profile. Okay. The myosin ATPase is in this flavor. And that flavor has more glycogen or less glycogen in it. Or it has more Krebs cycle enzymes in it or less in that way. And you begin to build up these profiles of that Fibers that have a slow myosin ATPase look like this. Fibers that have a fast myosin ATPase look like this. Okay? And that was how we initially classified fiber types. Okay? The more modern way of doing this, okay, involves something called immunohistochemistry. Immunohistochemistry. I will use a common thing to illustrate immunohistochemistry. Okay. How many of you guys have taken an at-home COVID test? Right? My guess is almost all of you. I hope almost all of you in those ways. If you've taken an at-home COVID test, okay, that functions with immunohistochemistry. There are antibodies that we have made that bind specifically to certain proteins or pieces of protein. Okay, the antibody only binds on to that. And when you do the COVID test, right, you swab your nose, you mix it with the liquid, and then it runs down on the chromatography paper. And if there's anything from that COVID virus that's on there, then you get the little line. Okay, the same thing happens when we do fiber types. It's just that we have antibodies to very specific pieces of the myosin heavy chain. We call it MHC. Okay, and that myosin heavy chain difference between slow twitch and fast twitch, and slow twitch, right? Or I'm sorry, in fast twitch type A and fast twitch type X. That's how we do. So we also have those now for in any kind of enzyme. We just have an antibody that will bind to anything in your body if we want. So we do immunohistochemistry now, and we're a little bit more accurate. I'm telling you guys all of this, not because it's super fun and interesting, but the how we arrived at these things controls why fast twitch fibers are called fast twitch fibers. And why, right, we had to switch how we name things. Now we call them type 1 and type 2, or where the letters and things come from. It all derives from how we used to do things versus how we do it now. Okay. So when we look at fiber typing, the original convention based upon the myosin ATPase, the histochemistry stain, was there were two types of muscle fiber. There was a fast fiber and a slow fiber. And that's solely based upon myosin ATPase enzyme function. Okay. So the original convention is fast, slow, just two. Okay, just two. And within fast and slow, then we can see who has more mitochondria, who has better glycolytic enzymes, right? Who's got more glycogen, et cetera, et cetera. So that's where fast and slow came from. Okay, that was first observed in animals, cats, dogs, rats, mice. The history of animal research is broad, but that's where it came from. What we know now, though, is it's a little bit more complicated. As we got better biochemistry techniques, we could figure out that within fast twitch fibers, there are actually some different flavors. Okay, slow twitch fibers, it's just one. Okay, but within the fast twitch fibers, there is this type A, this type X, and a very rare one in humans called B. B is very common in cats, mice, rats, lower animals that can run really, really fast. Okay. So we'll see about those. And then we now know there's a bunch of hybrid fibers that exist somewhere on a continuum in between all of these. Okay. So type 1 versus type 2, 1 denotes slow, 2 denotes fast. Okay. That matches with our old convention. And then there's the lettering portion. Most of you guys, the type 2 fibers you have are either A or X, or there's some kind of hybrid fiber that exists in between. Okay? A or X. If you're super sedentary, or you've been bedridden for a while, you've been in a cast for a while, or you have a spinal cord injury, we can begin to see a few type 2B fibers, but they're very rare in normal ambulatory humans. Okay? They're very, very common because they're the fastest ones. They're very common in lower animals. Okay. So what I'm going to show you here is this is a cartoon from a book trying to show you guys in the top left. This is what unstained fibers look like. Okay. And then when you do regular histochemistry, the old way of doing things, what you see here basically on the top right is a thing that will separate type 1 from 2A and 2X, okay? So you incubate it, you stain for the myosin to pH, you incubate it in an acidic environment, so a pH of 4.3. I have unfortunately done this before, and it basically turns those type 2 fibers white, they don't stain. Okay. Then you can see down here, right, same kind of thing. We're beginning, if we want to try to, we make it a little less acidic, and these things are gonna start to be, they're gonna start to stain a little bit. If you make it basic, you make the stain basic, then the type 1 fibers don't stain and the type 2s do. This is the old way of doing things, is you would bathe these things in different pHs and then look at them. Some poor grad student looks under a microscope and counts up the fibers. There's a circle around the fibers to figure out how big are they, what's their classification. Okay, that's how we used to do these kinds of things. So I have a picture, right? This is an actual picture of a thing. On the left, this is the ATPase. This is myosin ATPase stain. The lightest fibers are type 1. Okay, the couple of black fibers that you're going to see there, those are type 2A. And then the gray ones are type 2X. This person was pretty sad. Okay, and so you just go through and you count. How many fibers do I have? Which ones are which type? And that's how we get our relative percentages. On the right would be doing the same kind of thing, but we're actually staining for an enzyme from the Krebs cycle, for succinate dehydrogenase. The ones that stain darker have more mitochondria. They've got more SDH enzymes. They've got more mitochondria. You would do these two things side by side. These are not the same fibers, but you would do them side by side. You take these little thin slices, you can match all the fibers up, and you say, okay, I've got one that appears to be a type 1 fiber, and that has X amount of mitochondria. I've got this one as a 2A, it has more or less mitochondria. I've got a person who's highly aerobically trained. I got a person who's highly resistance trained, and what do these things look like across them? That's how we would learn and do all of our fiber type things. Okay? All right. So nowadays we characterize fiber type on myosin ATPase, the metabolic profile in those fibers, the size and conduction velocity of the motor axon, so the ability of the nervous system to connect to those fibers, as well as how many fibers connect to the same motor neuron. Okay, and we can figure out how much force they can produce. And so we can get this much more sophisticated picture of what happens. And what we know is that type 1 fibers or slow twitch fibers function a little bit differently than type 2A, that function a little bit differently than type 2X. And that ability of those things to do things differently helps us be able to be not only somewhat kind of short burst fast at things, but also to have a capacity for going a long period of time and to be able to very precisely control, which we'll get into later on this week, probably on Friday, some more of that kind of stuff. Okay, but we can do any and all of these things, and it has been done, and that's why we know to be an endurance athlete or really, really good, you need a lot of type 1 fibers. They have a greater capacity to have mitochondria and make ATP anaerobic, but they're slower. So your sprint speed is not quite as fast. Your ability to jump high, right? Not quite as good. You got a lot of type 2 fibers, you'll be a better sprinter, a better jumper. But you can't go very well, right? If you have a lot of 2X fibers, you got like 2 contractions in you and then you're done. Okay? That's okay. But this is part of why we can have people that run 100 meters in 9.6 seconds, but also can run, you know, a marathon in under 2 hours. They're not the same people. You can't really do both. Okay. The other thing I want to impart to you guys, all of you have some percentage of these fibers, type 1 versus type 2, in every muscle in your body. Almost every muscle is going to be very similar. Okay. So if you have 50% type 1 and 50% type 2 in your quad, you're gonna be close to 50-50 in the majority of muscles in your body. You didn't pick this. You can't change this. You can't change it with training or wishing or anything else. Whatever your regular kind of baseline fiber type is as a percentage is based upon your parents. It's genetic. Okay? Now, postural muscles, say your soleus, your gastroc, right? Your erector spinae, tend to be a little bit slower. They have a larger percentage of slow twitch fibers than say your quads or your glutes or your biceps or something like that. Okay, so if you're normally 50-50, maybe your soleus is 65-35. The slowest muscle in your body is your diaphragm. Okay, is your diaphragm. And it needs to be. If your diaphragm ever fatigued, can't breathe. Diaphragm has been contracting roughly 6 to 10 or 12 times a minute, basically since you were born. And it's never tired. Can you imagine if you were like curl, right? Can you imagine how small your bicep would be if you've been doing that for 20 years, right? Okay, but that again, anatomy, right, and parts function. Different functions for different muscles and different muscle groups. They're going to have slightly different sort of underlying anatomy and physiology. You can, with physical activity and training, convert type 2X fibers into type 2A fibers. And you can increase the number of mitochondria into A fibers and in type 1 fibers if you do aerobic training. Or if you detrain, they go down. Okay? Can't turn 2s into 1s. Most of the time, you can't turn 1s into 2s. Okay? We can't train myosin ATPase speed. Can't do it. You're either going to be a sprinter or you're not. You're born as a sprinter. We can train mitochondria. I can make every one of you in here with a little bit of basic training way better endurance athletes. I'm not going to make any of you a sprinter. You can work on form, you can do some squats, you can do some plyometric stuff and make you a little bit more explosive, but that's mostly just genetic, right? And if you want to be an elite distance runner, that's probably also genetic. But I can make every one of you able to run a half marathon if you're just willing to come out there and grind it out. It's fucking terrible, right? It's awful. But we can do that, okay? The other stuff we can't really change. Okay, so this is going to show us maximum shortening velocity. So how quickly can the myosin ATPase hydrolyze that ATP, right? Let us progress in the cross-free cycle and turn the muscle on to generate force. You see type 1 on the left is the slowest, right? The slowest shortening velocity. 2A is in the middle, 2X is out on the right somewhere. Okay? So that's speed. That's how we get these things. Okay. If you want to be an explosive athlete, run fast and jump high, you need more type 2s. Okay. So if we look at what you're going to see here on the left is an idea of something called specific force. And then on the right is something called power. We're going to do the power part first. Then we'll talk about what specific force is. But this is power. Power is force multiplied by velocity. Okay. Sorry, we do math sometimes. Who's had physics? Might have physics? Physics 1? Okay. We won't get much beyond physics 1. We'll do a little bit of physics 2 and talk about electricity at some point. But power is force multiplied by velocity. Athletes need power. If you want to run fast, jump high, generate momentum to knock somebody over or something like that, you need power. So you get more power by being stronger or by being able to contract faster. Okay. So type 2 fibers, because their velocity is greater, and as we'll see in a minute, they can actually curl their thighs, generate a little bit more force. They're going to get more power. Okay. They're going to get more power. Now, the left one talks about something called specific force. And you're like, what the hell is that? Specific force is take how big the fiber is, okay, take how much force that fiber can generate and divide those two by each other. Okay, how much force are we getting per size of the fiber? If the fiber is bigger, it can have more actin and myosin, therefore it can probably generate more force, right? But if we normalize force per muscle size, okay, then we can get an idea of, is there some inherent difference between 2A and 2X and type 1? Type 2s generate more force per muscle size, okay? This kind of normalization is really important in exercise physiology. For the longest time, we thought that women were not proportionally as strong as men because we never bothered to normalize strength to how much muscle you guys have. Okay? We never bothered to normalize your VO2 max to your body weight or your muscle mass. When we do all that, we're like, oh, they're mostly the same, right? We're just like, guys are bigger and they're stronger, so they must just be stronger than us just because they got more muscle. Okay? So this kind of a normalization is really, really, so there are some differences. You have more type 2 fibers, you're likely going to have a chance to be a little bit stronger. Okay, for whatever your body size is. You're going to have a little bit more power. You're going to be able to run a little bit faster, therefore, and jump a little bit higher. And I'm sorry, but that's just the way that it is. Okay. Okay. So I've got a little thing that's going to talk about, kind of go through and talk about what these things are. I want you guys to pay attention to this, but I'm not gonna spend a whole lot of time there. The one thing I do want to point out, because it's going to be really important for the next stuff that we talk about, is this idea of how many muscle fibers connect to a motor neuron based upon their fiber type. And what we know is that type 1 fibers, there's a smaller number of type 1 fibers that connect every type 1 motor neuron than there are 2A and 2X. So a neuron and all the fibers it goes to is called a motor unit, right? A motor unit. So type 1 fibers have small motor units, fewer fibers. Type 2As have larger motor units, type 2Xs have very, very large motor units. And this is unbelievably important for how we control force, how you are able to do coordinated movements, and how you're able to do things like write and type and walk, where you use the type 1 fibers over and over again during these low force contractions, and they don't get tired, right? So this piece of all of this is also unbelievably important for how we are able to function. Okay, so we'll revisit that a little bit probably on Friday. Okay, so I've got those, talked about that. Okay, perfect. While I switch everything around, this will be for us to move into sort of neuromuscular anatomy. Several of you guys have emailed and said, oh, well, that's not right. No, maybe he's thinking about it. Like, what just happened? I don't understand. What is it doing? Nothing makes me feel more like an old man than technology. I am really old man who yells at cloud. There it is. Okay. Several of you guys emailed and said you were having trouble accessing the YouTube channel. Is that, are all of you in that? Are all of you in that mode? Because when I do it and when Ty does it, we can link to the YouTube channel, but it pulls up like the playlist, and you just find the one that says Spring 2025. Okay. And then I don't have last Friday's video loaded up yet, but there should be one from last week. You guys just check that before we're done today, and we can try to figure out what all is going on. Okay. All right. So we spent a lot of time talking about muscle so far in class. Now we're going to talk about the nervous system and how we use that nervous system basically to connect the brain to our muscle so that we can contract the muscle when we want it to. Right. So again, most of this stuff you guys have hopefully seen already. Okay. So we're going to go relatively quickly through quite a bit of this. Okay. If we look at how the brain communicates with your muscles, we're going to start by talking about, okay, what we call an alpha motor neuron. You have your brain, you have the motor cortex in your brain. It's actually more complicated than that, but that's what we're gonna go with. Right, in the spinal cord. And then in the spinal cord, there are specialized neurons called alpha motor neurons. Okay? And so we'll look at where those things are going to be, but every muscle fiber in your body is connected to one of these alpha motor neurons that sits someplace in your spinal cord. Okay? So we have about 250 million, give or take, fibers, okay, in the body. You have somewhere between 400,000 and 500,000 motor neurons in your body. Okay, every fiber connects to one motor neuron. Every motor neuron connects to many fibers. Okay, so this is kind of the convention that we have. Okay. The one motor neuron and all the fibers that it connects to, that's called a motor neuron. When you need to move, when you need to generate force, your brain needs to communicate to a muscle. It does so one motor unit at a time. You don't think about it. That's how it functions. The brain sends a signal down the spinal cord, it reaches the alpha motor neuron. That one alpha motor neuron will depolarize, and from there, you will start to get force production. Okay? The size of the motor unit depends on fiber type. It also depends upon how many fibers are in a muscle. Bigger muscles tend to have more fibers. They tend to have more motor units or more motor neurons. They're going to go to all of that. That's not always the case. Okay. Muscles in your hands and your eyes. You have a bunch of little small motor units. That's going to be important from a function standpoint that we'll probably get into on Friday. Okay. All right. So let's talk about what the structure of a motor neuron is and how that helps us in our communication. What you see, this is a cartoon representation of a motor neuron, right? The part at the top, okay? That's what we call the cell body or the soma. Okay? Neurons are just cells, all they are. Okay? The cell body sits in a specific location in your spinal cord. Okay? And that location, as we will see, is determined by what muscle does it go to. Is that muscle an upper body muscle? Is it a lower body muscle? Okay? It sits in the spinal. Around the cell body are a bunch of little tiny finger-like, thread-like structures called dendrites. The purpose of a dendrite, okay, is, y'all are too close together, but basically, it would be like if you put your arms out and you're feeling around for where your neighbors are, okay? Or if you're waiting for your neighbor to give you like a high five or shake your hand. Dendrites exist to gather information from adjacent neurons, okay? Your spinal cord is just a collection of nerves, it's just a big collection of neurons. Some of them are motor neurons, some of them are sensory neurons, some of them are what we call interneurons, but they're just a bunch of cells. So dendrites gather information, okay? And they take that information and tell the cell body to do something or not to do something, okay? So they're bringing communication in, all right? The long structure, the thing you guys think about as a nerve, okay, is really the axon. So you have a cell body in the spinal cord, and then it spits out this rope-like structure that leaves the spinal cord and runs all the way out to the muscle that it's connected to. Okay, so that's the axon. What you guys think of as a nerve is really a bundle or a collection of axons that are all packaged together, right? So I have a nerve, right? This is the ulnar collateral, one of them runs under here somewhere, right? And if you were to do the dissection, right, if Cindy's making you guys name everything and being super tricky, you open up and you see what you think of as this is my ulnar collateral nerve. What that really is it's thousands of axons from all of the motor neurons and things, and then also there's gonna be 1A afferent axons that are coming up from stuff in your muscles. They all run together. Okay, that's what those things are going to be. Okay, that's the axon. On the axon, because the axon is long, okay? The axon tends to be wrapped in something called myelin. Myelin is like insulation. Okay, it's like insulation. It's a lipid, and what it does is it increases the diameter of the axon, and it provides sort of a cover over them so that we group our sodium and potassium channels all in little places that are called nodes across the end. But it lets us send signals faster. Okay? You imagine, right? I am not a particularly tall person, but I have motor neurons down here, right? At like L2 or something that are gonna go, or L3, they go to my feet. They control, right? The muscles that control my toes, right? So, I mean, that's cerebral feet that signal has to be able to travel. And we need it to go fast because God forbid, right, I step on a Lego. Okay? Step on Legos all the time. Right? You will one day too. You step on a Lego, or you step on something hot or cold, or some indeterminate fluid that's on the floor, right? And you don't like it, you want to pull your foot away. You want to wiggle your toes in a certain way. It needs to happen fast. And so we've got to cover that distance, okay, as fast as we possibly can. Right? And so that's why we have myelin. There are some diseases where what happens is the myelin goes away. And as the myelin goes away, you slow down. Okay? In certain places when you have multiple sclerosis, myelin gets worn away. Contraction velocity. Okay? My best friend had a thing when we were in college called Guillain-Barré syndrome. If you watch enough of sporting events and they got all of these fancy drugs, you guys ever notice if you watch like the national championship game, right? The only commercials are you like watching golf or something. All the commercials are for drugs of some sort, right? They're usually erectile dysfunction drugs or heart disease drugs or something to get rid of your eczema or your toenails or whatever, because those drugs work. We can't cure anything else. Those work. You guys know America is one of the only countries in the world where drug companies can actually like advertise on TV. In other places, you have to communicate with doctors directly. So it's good and bad to all of that. And those was just sort of a curiosity. But I had a point to all of this. One of them, they say it increases your risk of Guillain-Barré syndrome. Guillain-Barré syndrome, your immune system attacks the myelin on your muscle or on your axon. So my best friend, we were in college, and he went from normal, right? He was a pitcher on the baseball team, was fine, and then 2 days because all the myelin got eaten off his axons, he basically couldn't walk, couldn't move, couldn't do things. Okay? No idea why it comes on. Right? It basically filtered his cerebrospinal fluid and his plasma, like he was on dialysis. It's a similar thing. You try to rid of all of the antibodies, and then eventually the myelin goes back and you're fine. Obviously, this is why this is so important. Right? If you don't have myelin, you can still move sometimes, but it's going to be really slow, and that's not advantageous. All right. Couple of structures here in the cell body that I want you guys to be aware of. The first one is the nucleus. Okay, and so we're going to get into on Friday and next week talking about neurotransmitters and things that these nerve cells use to communicate with each other. Well, you need to know the nucleus because the nucleus is where we have the genetic information to say make more acetylcholine, make more epinephrine, or they don't. But make these neurotransmitters. Okay. In all of this, there's a thing called a nucleolus that you guys can see in there. That's a collection of ribosomal RNA, which is going to help us make proteins. So the nucleus is really important because it has to make all of the things that this nerve needs to stay healthy. Okay. There's a thing called Nissl bodies, which are little collections of endoplasmic reticulum and high concentrations of ribosomes. So we make the proteins right in those ribosomes, we package them up in the endoplasmic reticulum, and then we basically walk them down the axon to get them down to to the muscular junction. Now, walking down is done by these structural proteins, that are, we call cytoskeletal proteins, they're gonna be in the axon. Microtubules, there's actin filaments, there's what we call neurofilaments. All of this stuff provides scaffolding and an ability to walk stuff down. Okay, as you all sit here, you are walking proteins from in your spinal cord down your axon. Okay, you have these motor proteins, they're like myosin, but there's one called dynin, there's one called kinesin, and you hook little vesicles on them and they walk their way down. Without all of this, without the microtubules, without that structure, okay, you could not regenerate an axon when it gets damaged. You could not repair your myelin when it gets damaged or something like that happens. If a cell body, if the soma in your spinal cord dies, that whole thing dies. But if the cell body is okay, I can cut an axon, okay? I can cut the axon. And it will grow back, as long as it still has those kinds of things. I had a buddy, that's what he did for his postdoc, was he would cut axons on like rats and mice and things, and they would make them walk on a treadmill afterwards, and it helps to regrow the axons better. And so that's why all of this stuff is so important, right? Some nerve damage, as you guys age, you'll have nerve damage. I lack feeling in the tip of my finger. I have impingement and some things here at C4, C5. We'll talk about that later. It's fucking great to be old. You go to sleep one night and you wake up and all of a sudden, right, you're like, I can't move. My neck hurts in a way that you've never thought before. Your bicep is numb right down through here. Dermatomes. Yeah, I still have some of the stuff, right? It's a cautionary tale. Don't smash your head into people playing football when you're 14, when 15, 16, 18, and in the midst of all of those. Probably too late for all of us in the midst of that. Okay. Okay. So how am I doing on time? Are we getting close? Yeah, I got a couple of minutes. So we'll finish with this. Okay. I told you guys before that motor neurons are located in very specific places in your spinal cord, and they are grouped together. So like all the motor neurons that go to a specific muscle are in a very, very similar place in your spinal cord. All right. And so if we look at all of this, right, on the right, we've got kind of a sagittal cross section of the spinal cord, and you can see all of the different vertebrae. So it starts at C1, we're moving all the way down here to S5, okay? And so like at C3, like the highest ones, the diaphragm is the highest, okay, closest to your brain. Most of the rest of them are going to function though kind of in order of as you move down the body, we're going to move down. Why do you guys think, why would it be useful for the diaphragm to be as close to the brain as possible? This is the fun part that everybody hates. All right, Cooper put his on, so I was going to call on somebody. Would you like to call? No, it's okay, go for it. Speed. Speed, okay. It's closer, I can get it to contract faster, that's good. Why would it be important to contract my diaphragm quickly? So you can breathe. Art it. Okay, there's just kind of an optical illusion on your thing. You gotta be able to breathe, right? So we'll see if you get damage somewhere in the spinal cord, nothing gets past, and so it's advantageous for the diaphragm to be up there a little bit higher. Okay, but right. You look at all of this, C5, deltoids, biceps, right? Shoulders, biceps, wrist extensors, weirdly triceps are lower. That's a strange thing. Hand muscles in the midst of all of that. Okay, those are going to be in the cervical vertebrae. You have almost no motor neurons in thoracic vertebrae. And then most of our lower body ones are going to be down in the lumbar. Okay, hip flexors, knee extensors, dorsiflexors, plantar flexors are going to be, down in the lumbar and into sacral. Okay. Having them grouped like this has advantages and disadvantages. Okay, and that will be where we will pick up on Friday. Okay, that's where we'll pick up on. Okay. Please throw your straps away. Please make sure if you have a marker that you return the marker so we don't get the payback from Ellis. And please hang on to and bring back your nameplate in the future. Okay? All right, we'll see you guys on Friday.
So I know I'm thinking way ahead, but I remembered that there was a graphic you can use for like policies.